Up next is introduction. Mendel's first law of segregation. This law states that during the formation of gamete, each gene separates from each other so that each gamete carries only one allele for each gene. So, from the diagram, each gamete P of P generation should carry their own allele. For example, a green allele and a yellow seed strain. Next, we have the F1 generation, which is the hybrid. Here we can see that the characteristic of the plants fully follows the domain allele from the P generation, which is yellow. And finally, the last generation of F2 generation. Here it follows the ratio 3 to 1, where 3 domain allele and 1 recessive allele. So we got 3 yellow P's and 1 green P's. Up next is the relation of Mendel's first law to discrete math. There are 2 chapters from discrete math which was applied by this law. One of them is chapter 2's logic and proof, specifically truth table of conjunction. Firstly, we were to let y, capital letter Y, as the domain trait and lowercase y as a recessive trait. Then we will insert them inside the truth table of conjunction, and there is an and if you notice from the last column, P and Q, if there is at least one capital Y in the table, then the trait should be follow the domain trait. Up next is the chapter 6 probability, where according to Mendel's law, the ratio between domain and recessive trait is 3 to 1. So first, the event where first of all, we have to find the probability of seed being round, which is a domain trait. So the event should be equal to 3 according to the ratio given, and the sample size is 3 plus 1, which is 4. Thus we just divide. According to the formula, probability of the event is equal to event divides by the total events. So 3 to 4. Thus the probability of the seed being round is 3 to 4. In short, discrete math is used to calculate the possible outcome of plant trait in Mendel's theory. Mendel's segregation law.
Now we will go to the history of the contribution. Mendel began his education as a priest at the Augustinian St. Thomas Abbey. He began his research on heredity using mice, but his bishop did not like one of his friars studying animal cells, so he switched to plants. In a monastery's garden, Mendel planted and examined roughly 29,000 garden pea plants and analyzing 7 traits of the garden pea plants. The 7 traits of the garden pea plants are flower color, which is purple or white, seed texture, which is wrinkled or round. Next is seed color, which is yellow or green, and stem length, which is long or short, and pod color, which is yellow or green, pod texture, which is inflated or constricted, and lastly flower position, which will be axial or terminal. Based on the appearance or phenotypes of the 7 traits, Mendel developed genotypes for each of those traits. The fundamental principles of heredity, known as Mendel's laws of inheritance, were revealed because of Mendel's work. We now understand that genes, which are carried on chromosomes, are the fundamental functional units of heredity, capable of being reproduced, expressed, and altered. Mendel's postulates are the foundation of classical or Mendelian genetics today. According to Mendelian genetics, not all genes are passed down from parents to children, but Mendel's experiment provide a wonderful place to start thinking about inheritance. All of Mendel's observations and discoveries were made by crossing individual plants. We can now see a human chromosome abnormality which contains all an individual's chromosome to detect chromosomal problems in offspring even before they are born. Other researchers noticed that distinct traits were frequently inherited together shortly after Mendel hypothesized that features were determined by what are now known as related by being situated on the same chromosome. Many of the breakthroughs in molecular biology have been traced back to Mendel's work.
Today, I will talk about Morgan's work on supply, or we can call it as Drosophila. So, from the picture shown in the slide, you can see a scientist named Thomas Morgan. He was born on 25 September 1866 in Lexington, Kentucky. He worked on Drosophila, where he crossed homozygous long-winged Drosophila with homozygous vestigial wing Drosophila. Now, we move on to the process of segregation during the cross between long-wing and vestigial wing Drosophila. So, we suppose the long-wing Drosophila has a pair of V plus alleles, and for the vestigial wing Drosophila has a VV alleles. A gamete with a single V plus allele is produced by long-wing Drosophila, and a gamete with a V allele is produced by vestigial wing Drosophila. So because V plus is the dominant allele, which are presumably seen as long-wing Drosophila, it produces long-wing heterozygous Drosophila. So as you can see from the diagram, it shows how alleles are segregated during gamete production and combined to make three different genotypes in hybrids. Relate our experiment with the subject discrete math, I make a table and probability calculation for the cross between long wing and vestigial wing. You can see from the table there's two different alleles, which is V plus and V. So from the combined allele we can see three long wing Drosophila was produced and one vestigial wing was produced. Move on to the probability calculation. We can assume the ratio is 3 to 1. So the total of long wing Drosophila is 3. Total of Drosophila that produced from the combined allele is 4. So we can assume the probability of Drosophila being long wing is 3 over 4.
Another application of Mendel's law in real life is a study by William Jasper Spillman on practical problem in breeding cattle. William Jasper Spillman is known as the founding father of agriculture economics, notable for independently rediscovering Mendel's law of hereditary. He conducted various studies related to breeding and hereditary in hybrid offsprings, and one such example is his study on several herds of old Hereford cattle in an article titled Application of Mendel's Law to a Practical Problem in Breeding Cattle. In his study, the pure pork cattle is represented by P and the horn cattle by H, as seen in the table at the top left. Each of the H ovules is offered both P and H gametes equally, so the chance between PXH and HXH is even. The Mendelian expectation for this study is as written by Spillman in the middle left. The table on the bottom left shows the data captured during the past season when the study was conducted. From the table, it can be concluded the expectation is realized or very near with few exceptions.
Okay, going on to the next part, we'll be presenting about the impacts on human life. There are two impacts that we came out with during our research study, which is first of all is the RH incompatibility during pregnancy, and the next one is the inheritance of thalassemia. Okay, so what is RH factor? RH factor is also called a rhesus factor, which is a type of protein that surrounding the blood cell. So the protein is genetically inherited from our parents. So if you have the protein, you are Rh positive. If you didn't have the protein, you are Rh negative. So there are two types, positive and negative. Have protein, positive, and didn't have protein, negative. So most people in the world are about 85% RH positive. Okay, just now we found out about the rhesus factor. Now we need to know about the RH incompatibility. So RH incompatibility occurs when a RH negative mother exposed to RH positive blood from her son or her child, resulting in the development of RH antibodies in the mother. So why this happen? When the fetus came out, it is considered as an alien from our body because it contain RH positive blood, which is different from the mother RH negative. Just like COVID-19, which is an alien form of virus, when entered our body, so it's triggered our self-defense system, which resulting in the development of antibodies. So when this happens, the antibodies will attack the red blood cell, which will cause hemolysis or the destruction of the fetus red blood cells. So when this happens, it will become anemic as a result, a blood disorder in the fetus. So the most common form of RH incompatibility occurs when a RH negative mother and a RH positive father produce a RH positive fetus. Okay, next we will try to apply the Mendel's first law of segregation to predict the RH incompatibility during pregnancy. As we can see in the figure, the mother and the father blood rhesus factor, which is for example, if the mother is negative and the father is negative, their child will be negative. So as the ratio shown, the ratio will be 3 to 1. The event will be 3 in the event which is a positive child, positive rhesus factor child. So probability of the child's RH factor negative is 75%.
What is thalassemia? Thalassemia is a hereditary blood illness characterized by the body's inability to produce enough hemoglobin. That is a key component of red blood cells. All the blood cells receive oxygen from the red blood. When there aren't enough healthy red blood cells, a person feels very weak or short of breath. Anemia is the medical term for this ailment. Anemia can be minor or severe in people with thalassemia. How can we detect a person with thalassemia? We can use the Mendel's first law of segregation. This picture shows a karyotype or a picture of an individual's genetic information as seen via a microscope. There are 22 pairs of numbered chromosomes, autosomes, and 1 pair of chromosomes that determine sex. Also, a male possesses both an X and a Y chromosome, whereas a female possesses 2 X chromosomes. Next, we will talk about beta thalassemia. Most persons with beta thalassemia have mutations in both copies of the beta globin HBB gene on chromosome 11 in each cell. The parents of an infected person are known as carriers, since they each have one mutant copy of the gene. Although some carriers of beta thalassemia develop moderate anemia, most carriers do not display any signs or symptoms of the disease. When 2 autosomal recessive disorder carriers have offspring, each child has a 25%, that is 1 in 4, chance of having thalassemia, a 50% 1 in 2 chance of being a carrier like both parents, and a 25% chance of not having the condition and not being a carrier. So I think that's all from us. Thank you.